Introduce Dr. Steve Perrin, who is CEO and CSO, Chief Scientific Officer of ALS Therapy Development Institute. He's an expert in drug development and the process, and has had nearly 2 decades of experience leading preclinical and translational research efforts. He's assembled a team of more than 30 full-time professionals to execute projects based on information that the Institute itself has developed. And I'm sure we're all looking forward to hearing about the future and precision medicine and telemedicine. Dr. Perrin? What's it? Yeah, it's not heavy. I just.
It is really heavy.
It's not that heavy.
While this is loading up, I should say thank you for inviting me to present today. It's actually great that Jonathan, James, and I did not actually talk to each other about what we're going to present, because I think I'm actually going to fit in the middle of the two, and unfortunately I'm throwing Terry a little bit of a curveball on my title, which is okay. But let me see if I can get this thing going. There you go. So I'm going to talk a little bit about telemedicine and telehealth, but I'm not going to talk about the future of it. I figured it might be more interesting to try to tell you how we're actually applying it today in the context more of drug development, and kind of tie in some of the things that both Jonathan and James talked about. So what is telehealth? Let's start there. It's not a new term. It's probably first pops up in the literature in the 1940s, 1950s. And it's not unusual when you say it actually was started and funded by the Department of Defense. And initially, it was really designed to try to bring health to remote areas. People living in the desert, hard to get to clinics, how could they communicate with healthcare teams? And of course, they had a secondary utilization for it, military context, obviously, but also NASA was interested in it because sending people to long-term space missions, living on the moon, et cetera, they wanted to develop tools and mechanisms for people to be able to access the healthcare system here back on the ground, if you will. But looking at what that means to us today, I think we're seeing the emergence of disciplines happening in real time in the last decade or so. And Jonathan talked about a really great example of precision medicine technology applied clinically, if you will, with proteomics. Even 10 years ago, technologies around precision medicine really weren't available to clinical studies. It was cost prohibitive. The first draft of the human genome cost billions of dollars and took 10 years to develop. Even when the technologies got cheaper and the throughput was higher, it was costing tens of thousands of dollars per sample to do a study like Jonathan described in his presentation. And today, you can sequence a genome, probably $500, takes a couple of weeks. The analysis of the data is challenging, but the cost of the technology is to look at the genetics of diseases, to look at the biomarkers at the RNA and protein and even cellular level, and even at the single cell level, didn't exist 5 to 10 years ago. And if they did exist, they existed in very few labs, and very few people had access to those technologies, whereas today, it's pennies on the dollar and you can actually include them in clinical studies. And that's why these fields are merging. So when you hear the words telehealth, telemedicine, precision medicine, translational medicine, it's not about the clinical team picking up the phone and calling a patient about a clinical visit. It's about applying really powerful tools and technologies to disease. And the downsides, it generates a ton of data. So why are we doing this? And James really set this one up quite well for me in his talk. And this is ALSFRS scores from our precision medicine program over the last 2.5 years or so. But this slide is no different than if you look at ALSFRS in a general ALS cohort that has not been stratified or reduced heterogeneity, whatever terms you want to use. This is how variable progression can be. And that's really challenging when one thinks about that in the context of drug development. So again, a fairly old piece of data, but a power analysis on exactly what that means in the context of drug development. In this particular example, to detect a 25% change in ALSFRS requires about 450 participants. Even though James is accurate when he said We typically don't want to measure disease progression in a phase 2. He was being really nice. If you go into clinicaltrials.gov and look at clinical trials back to 2000, 85% of the trials uses it almost as a primary endpoint, and they're powering them at 100 people per group. There's no way you could measure efficacy in any of those trials, period. So it's a problem. We need more unbiased quantitative outcome measures to design clinical trials in ALS. And ALS is not the only disease that has these issues, but because of some of the things you've already heard today, it's a little bit more challenging to measure disease progression in ALS in a very quantitative way. So what does this mean in the context of drug development, and how is precision medicine or telehealth going to help us with this really big problem? It helps us all across the value chain of how drug development's done. And actually, I guess I wish I would have talked to both of them before, because I'd fill in some of the details here a little bit more. So I may pause on certain slides and go off topic just to try to fill in some of those details. But precision medicine and telehealth can impact the entire value chain, all the way up to understanding how a disease works, what are the targets that may help us slow down the disease if we can effectively develop a drug, help during the drug development process to pick the winners, and most importantly, as James said, as I just alluded to really optimize the design and interpretation of your trial once you get there. Because we can't use ALSFRS from an economic standpoint in all of our trials. It's cost prohibitive. It would take 400 patients per arm in a multiple dose study. You're talking hundreds of millions of dollars, and the average small biotech company or a VC group that wants a quick return is not going to invest in a trial design like that. They want a shorter term outcome than what we just described. So back in 2010, we decided that we wanted to start to grapple with that at ALS TDI, and we wrote this huge grant to the NIH. It was what they called the Transformational Grant Program, 5-year grants, $10 million per year. We were kind of piggybacking on what had been published in oncology and precision medicine, said, hey, we want to do that in ALS. It got the worst grant scores I've ever seen. It was really horrible. But because we tend to take no for an answer, we went and hired the head of translational medicine at Biogen at the time in their immuno group, Matvey Lukashev, hired him in 2011 or 12 and said, hey, build a team. We want to do precision medicine in ALS. And he came on board, which was kind of surprising to me. But he came, he joined, he was really excited. He hired a 5 or 6 person team. He said, what do you want to do? I said, well, let's see if we can get people with ALS to come to the institute or to a local clinic. And we don't know what we're looking for, so let's do a fishing expedition. Let's collect longitudinal biosamples, blood in particular, easy to get. Let's sequence their genome, full 30x coverage. It's only $2,000 a piece back then. Now it's under $1,000, as I said. Let's sequence their full genomes. I don't know why, we might find something. Let's look at biomarkers in their blood, unbiased. Let's look at their proteome, their RNA for as many analytes as we can look at. Let's look at the various cell types that are in their blood, and let's follow them over time. Let's look at clinical endpoints. Let's ask them on a monthly basis to provide us with a self-reported ALSFRS. Let's ask them to provide a voice recording every month. Let's send them home with accelerometer devices on their wrists and ankles. I know that that looks weird, and actually if you've seen pictures of people wearing them, they look like they're in prison, but we asked them to do it anyway. And we asked them to do that because of the way the disease spreads. James alluded to it. Not every single patient progresses in the same way or at the same rate. We didn't know what we wanted to measure, so we asked them to wear them on all 4 limbs, and that would give us better data than an app on a phone that you're wearing on your belt. That was our idea. The devices are also exquisitely sensitive. We didn't promise any treatment. We didn't promise a cure. And yet from around the globe, we had 485 people come to Cambridge and enrolled in the program since September 2014. It generates an incredible amount of data. Jonathan alluded to the fact that these types of technologies generate large quantities of data, and that is problematic as a whole. But it's been a great program. I'll share a few things with you. I threw a few demographics in here, and again, not on purpose. This was not one that we tried to randomize, like James said. It was all comers. If you could get to Cambridge, especially in the first year of the program, we let anybody come in. We've kind of shifted a little bit to people that are newly diagnosed in the last year or so, and we've had some reasons to do that. But in 2014 and half of 2015, if you could get to Cambridge on your own dime, you're in the program. We're going to sequence your genome. We're going to make an IPS line from your skin. We're going to give you devices. We're going to set you up with a web portal. We're going to sequence your genome and share the data with you. So it wasn't balanced. We had a lot more males show up than females. Don't really know why. The age balancing was actually pretty good in the cohort across, heavily Caucasian, as you can see. We did include a fair amount of healthy volunteers in our study as well. They got their genome sequenced for free. They were very excited about that. Genetic mutations, pretty well covered. You heard about some of them today, and as you saw from Jonathan's data, C9orf, pretty common, was probably the most common one that we've seen so far. These data actually might be a little bit outdated. I think we're up to 28 or so C9orf positive people in the cohort. SOD, pretty common, but that's because we targeted some families, so that's why that one's a little higher than what you might expect. And then some rare mutations that were on Jonathan's slides. One of them I'll mention just because one of the stories that he told, and that's the two folks that enrolled in the program that had mutations in DynAct and heavy chain. Interesting gene, axon transport is its biological function. The mom has the mutation, full-blown cognitive dysfunction, absolutely no motor dysfunction whatsoever. She's in her late 70s. Son, no cognitive impairment, completely actually passed away recently in the last year or so of ALS, no cognitive dysfunction that the clinician could find. So it shows you that spectrum again. One of the things that is interesting about this, and again, I alluded to it and Jonathan mentioned it with C9orf, we have IPS lines going for most of these types of mutations at this point, and how we apply those to the drug development process is going to be quite interesting over time. So what about compliance? And we talked, I think James mentioned compliance once. It's a really ugly word. Nobody likes to talk about ALS clinical trial compliance. It's about a 20% drop-off in most trials, and that's because going to a 12-month study with lots of clinical visits is really challenging as folks progress with the disease. And again, if we can start to use telehealth, tools that you can measure things at home without having to do a clinical visit, should boost compliance quite a bit. And we've seen that a little bit in our program. The folks in the program only had to come to Cambridge once, and that was mostly to get the skin biopsy and to get the first sets of blood samples that we wanted for genome sequencing stuff. After that, everything was done remotely. They had a web portal to do their monthly ALSFRS, to provide their voice recordings, and we shipped the accelerometers back and forth, download the data, erase them, and send them back out. I won't tell you why we didn't Bluetooth the data, but there were some regulatory reasons why we chose not to. The interesting thing about compliance in our program, the dropout rate was about 20%. Out of the entire cohort of about 485 participants, 80% of them gave us at least 3 months of data from all of these outcome measures. And if you tended to do one thing, you can see the big circle window, you tended to do all of them. About 35% of the participants, if they were giving us monthly ALSFRS, they were doing all three pretty compliantly. You can see there's a little bit better compliance between ALSFRS and accelerometers compared to either one with voice. Not really so sure why. One might hypothesize that everybody knows that ALSFRS is used in clinical trials as an outcome measure, therefore they were really regular about doing their ALSFRS monthly. Maybe they forgot to just click on the next web page, portal on their web page and do the voice recording. Clearly accelerometers was the most challenging. It's the one during disease progression that we saw the steepest drop off. It fell off after about 18 months, and that's because it was really hard to do some of the exercises that we asked our participants to do. We are not the only game in town doing precision medicine. There's lots of them cropping up. Everybody's doing a fantastic job with what they're doing. None of us are doing it the same, which is a little bit unfortunate. Some people are using accelerometers, some people are using apps, some people are looking at epigenetics, some people are not doing whole genome sequencing. But the bigger challenge that we're not all doing it the same is none of us are sharing our data. If we really want to leverage telehealth in the future, we have to figure that one out, because what we really want to leverage is baseline data for people before they get their horrible diagnosis. Often it takes, as you all know, 3 months, 6 months, 9 months to get a diagnosis, but often you've already been in the clinic and there's some hint that somebody thinks you might have ALS. If you can get them into a program like this prior to their diagnosis, you're going to have a lot of information about how to stratify them, what their progression rates might be, what their fluid biomarkers look like. Are there genes in their genome that could help stratify them as fast or slow progressors? These are all things that could happen very, very quickly within a month or two of showing up in the clinic for even getting a possible diagnosis. And we're not doing a very good job as a community with that piece of telehealth precision medicine today. So how do you utilize this type of information for drug discovery? And does it have an application there for identification of new targets, new biology? And the answer is yes. An example of inflammation, something near and dear to our heart, but I figured I could do a little bit of follow-up during the presentation on how we applied it, is we went and started looking at immune markers in ALS patients' blood back when I joined TDI in 2007. It was one of the first things that we did. I think we enrolled 600 people, collected their bloods. We're using what was back then the cutting-edge technology that today is way out of date. But we found that the co-stimulatory pathway, a specific immune pathway, was highly upregulated in about 70% of patients in the cohort, which was quite striking for us. We immediately jumped into a mouse model, the SOD1 model, which is the most common model used. And Jonathan hinted at one of the reasons why. It looks the most like ALS. It has a lot of the pathologies that ALS has when we see in patients. Many, many other genetic mutations that we talk about, the animal models that we use, and I don't care if they're rats or mice, they don't really show the same things that we see in patients. And in fact, there's probably 20 TDP43 animal models that have been made in rodents. Only one of them might show what really does look like progressive neurodegeneration. There's been about a dozen CN-ORF models made in rodents. We finally might have one that looks like progressive neurodegeneration. So making an animal model of the disease is really hard, even if you know the genetics. And this is not unique to ALS. When I ran Biogen's biomarker discovery programs and I worked on models of multiple sclerosis, lupus, cancer, none of them look like the human disease. They're tools. They have hints of looking like the human disease, and you can use them as a tool to say, geez, this pathway changes in the model. It also changes in the human disease. If I make a drug that targets the pathway, does it do any good? And that's really what the starting point of that was. When we found that co-stimulatory pathway, we went and looked in mice. And lo and behold, when you look at the genes in that pathway in a mouse, the SOD1 mouse, the pathway increases longitudinally over time in almost every tissue you look at that's relevant to the disease. Spinal cord, blood, brain, sciatic nerve. The pathway starts at symptom onset and just increases over time until the animals basically die. So what was interesting about that is that other groups using different rodent models and thinking about the disease in those models a little bit differently were finding similar things that overlapped with the co-stimulatory pathway. That pathway is expressed on lymphocytes and antigen-presenting cells. Antigen-presenting cells comes in lots of different flavors depending upon what tissue you're looking in. They could be monocytes, they can be microglial cells, they can be sensing cells in the skin and in the gut. Any cell that expresses co-stimulatory molecules that talk to lymphocytes on their cell surface is an antigen-presenting cell. Stan Appel was looking at those types of cells when he was crossing SOD mice into mice that lacked specific lymphocyte populations, and lo and behold, he was surprised. When you totally knock out lymphocytes, the disease gets worse, meaning, gee, something in the immune system, even though it goes up, is pretty protective. He then did a second cross where he looked at a specific type of receptor on lymphocyte, CD4 positive ones, and again, showed that if you knock them out, the disease gets worse. So something in that subset of lymphocytes was interesting, and if you completely got rid of them, you actually made disease worse. And another different lab was taking more of a, what I guess I'd call a pharmacological approach and said, well, geez, if we put a specific cell type of lymphocytes back in, does it make disease better? And those were Treg cells, and the answer to that was yes. It was a small effect, but it looked fairly reproducible. Mikhail Schwartz, on the other side of the pond, if you will, was doing some other pretty interesting things that overlap with the co-stimulatory pathway as well. And again, mostly with transgenics, but a little bit of pharmacology. She induced an autoimmune antigen response, if you will, by treating the animals with a peptide called MOG and really slowed down disease. And this really boosts cells of the Treg lineage, if you will. Now, it's nothing you'd ever want to bring into the clinic. You don't want to induce multiple sclerosis in people that already have ALS. That would be a bad idea. But what she showed in the mouse was really telling, because when then she looked at the cell types that were getting into the brain, when she gave this challenge, they were the exact cells that Stan was looking at. Tregs and cells of the monocyte lineage that express high levels of IL-10 that knock down and slow down pro-inflammatory signaling. So how does this relate to co-stimulatory pathway? Because that's kind of how I opened up how ALS TDI got interested in the pathway. Was, 20 years ago at Biogen, we put a drug into the clinic modulating co-stimulatory pathway. We didn't even know how it worked back then. But we knew in animal models that if you gave them really high doses and really knocked down co-stimulation, It really helped transplant. You could do much better with transplant if you knock down the co-stimulatory pathway. We didn't have a great idea back then why, but that literature starting to unfold. And how is tolerance induced? By introducing Tregs. It upregulates Treg populations very highly, as you can see, and they express incredibly high levels of IL-10. So back in 2008, when we found those two pathways that I showed you, we decided, hey, you know, we do drug development in the sod mouse, so let's bring in the murine antibody that blocks the co-stimulatory pathway. We'd all used it at Biogen in multiple sclerosis lupus model, so we knew how to make it. Lo and behold, it was the first drug out of, I think back then, we had tested 200 compounds in the animal model. Nothing had worked in our hands yet. It was the first drug that in panel B slowed down body weight loss. Panel C, it slowed down the neurological disease progression in the mouse that we can semi-quantitatively measure. And most importantly, improved survival by about 20 days. Not a huge amount. When people tend to look at this type of data in our field, they go, oh crap, who wants to invest in ALS? A 20-day effect? You didn't cure the animals. You have to realize to make this mouse sick and get what we think looks like ALS, if you put 1 copy of the transgene in a mouse, they don't get sick. If you put 4 copies in, depending upon the background genetics, the animals tend to live 200 to 300 days. Not a good tool for drug screening if you want to cycle through lots of drugs. This animal expresses 24 to 30 copies of that mutant transgene to make them sick. This is a pretty good effect in a preclinical rodent model, and it's reproducible. You can do dose escalation. It did lots of other great things too, though. When we did immunohistochemistry and looked at things that we know are going wrong in ALS, things like loss of motor neurons. The 2 right-hand panels there, the dark cells are motor neurons. Treat animals for 50 days, get them on drug, and then harvest the spinal cords. A huge increase in motor neuron survival. When the muscle atrophies, it's due to loss of the neuromuscular junction, or the place where the nerve connects to the muscle. When those nerves are detached, the immune system recognizes that, and macrophages, an immune cell, go in there and they just chew them right from where the muscle is all the way up towards the spinal cord. The green cells are macrophages. You can see the huge number of them on a nerve in the calf muscle in that top panel. It almost eliminates the macrophage recognition of that denervated nerve. And the actual connections, the actual neuromuscular junction connection itself, you can see in the graphs over here how many neuromuscular junctions are lost over a period where the animals look pretty healthy at day 70, first signs of symptom onset, to day 85, where their hind limbs are getting pretty affected. Massive loss of neuromuscular junction attachment. When you treat with the drug, it doesn't quite bring them back up to normal counts, but it does a pretty good job. And finally, if you look at immune molecules that are in the spinal cord without treatment and then with treatment, you knock down pro-inflammatory signaling, i.e. microglial activation and astrocyte activation, quite profoundly in the mouse. So it does lots of great things. So this is a typical, what I would call preclinical data package that one should have before you even think of putting a drug into the clinic. Clinical success in ALS is about 0.1%. It's not great. We just got our second drug approval in 20-something years. And that's true in the preclinical space as well. I think we're up to 380 drugs that we've tested in the sod mouse, and only 5 have worked. So we're at about 1%. So before you bring a drug from testing in cells in a dish, or if you choose to go through an animal model, at least have some basic checkpoints in place, or your likelihood of translation and success in the clinic is pretty slim. Now, good preclinical work like I just described, that was a couple of $1 million of work. It wasn't something that happened overnight. But based on a program like that, we got excited about actually bringing an anti-CD40 ligand antibody to the clinic. So we went ahead and we humanized it. And God, my marketing person, Colette, is cringing over the color translation. Sorry. I cut and pasted from somewhere else. But this is a human version of that antibody. Again, we started off with a murine antibody to do all the rodent work. You can't take a human antibody and test it in mice. That's a whole different phase of drug development. And that phase costs a couple $1 million. Well, to actually go and humanize this antibody, and then as a nonprofit to go out and hire a company that's going to establish a cell line with FDA principles to make enough protein to actually go to the clinic, to set up a purification strategy on that cell line, to test to make sure that it doesn't have any toxicity in non-human primates, to make sure that you can put the drug in its proper stable form so you can ship it to the clinic, to get it ready for all the regulatory paperwork. Geez, we started this process in 2010 10 or 11. We're going to have it in Q1 into phase 1. That's been about $9 million, and we did it really on the cheap. I'm trying to put some context of numbers here to show you that just because you identify a target, that does not give you something that's going to work on a phase 3 clinical trial. There is an awful lot of work that has to happen in between. So James mentioned phase 1. I'm going to skip over it because there's not lots of bells and whistles there. You're really trying to show safety, as he said. But phase 2 is really where the death of drug development happens for most diseases, and ALS is no different. First of all, it's cost prohibitive. You want to do dose escalation studies of some kind or other usually. You don't really know if your rodent doses are going to work in people. You don't know if the lifetime of your drug in circulation is going to be the same in rodents as it is in people or non-human primates for that matter. So you tend to want to do multiple doses in those studies. Think about the power that you need in an ALS trial without biomarkers that help you identify target engagement, whether you're modifying the disease. And sometimes disease modification is really easy to measure. Anybody that knows anything about psoriasis, really easy biomarker to understand whether your drug is working in a clinical trial. Psoriasis patients are covered with plaques on their skin. You can look at their arms. If you're giving them a drug and it's working, the plaques go away. Now think about that, how you're going to do that in the context of something like ALS. It's really hard. So the more that we as a field help develop more quantitative tools, not only for clinical endpoints, but to also measure quantitatively whether drugs are working, the better off we're going to be at translating drugs out of the lab and into the clinic. And James already used another great example here. This is examples of neurofilament levels that one can measure both in CSF and blood. Again, thinking about how we're going to transition our immune modulatory drug into the clinic. You can think about things like Stan Appel's doing. He took his information and his data that he generated in the SOD mouse with his Treg cells and has now correlated that your baseline levels of those cell types can help predict whether you're a slow or a fast progressor. Powerful tools when you think about the concepts that James was bringing to you about patient stratification in homogeneous groups. One of the other things that we're really lucky about, because this is an immune modulatory drug and we can measure its effects in blood, is that we have great biomarkers that are dose dependent that we can bring from our mouse studies and measure those in people so that at least when we're in our dose finding studies in phase 2, we'll know that we're measuring that the drug is hitting target and that it's doing what we think it should do. So to kind of sum up, the fields have merged. It's actually not even the future, it's today. The consumers in the world, including this audience, are utilizing technologies in a way that we couldn't have even imagined 10 years ago. They're taking their health into their own hands and they're making decisions, and we need to embrace that as a community. The cost to do technical assays that we couldn't have imagined to a decade ago are now here today, and they're being applied. There's biosensors that measure the levels of these types of analytes in fluid like saliva, sweat, and blood are here today, and they're going to keep getting miniaturized, and you're going to be able to buy them on Amazon, and consumers are going to buy them. So we as a community have to learn how to embrace that. I could thank a bunch of people at TDI, but I think because I talked a lot about our precision medicine program, the only folks that I really should thank is the ALS community, because again, they came and enrolled in our program with no promise of a drug treatment, no promise of a cure. They did it because they wanted to do something to impact the whole process of drug development, and we can't thank them enough for that. So thank you for your time.